So when it comes to genetics, it's really important to wrap your head around the terminology. So you might not be there yet in the course, but it's still no problem to learn terminology, and it means when you do get there, then it's a friend. You've heard it before, it's not brand new. Okay, so let's get going first with some genetics terminology. Right, so we are going to start with what is an allele. Okay, so an allele is a version of a gene. Remember, we've talked about DNA, and DNA has all the instructions to make every organism and to do every function in every cell. All of those functions and everything that the cell does is controlled by proteins. So the instructions for the proteins are coded for in the DNA, and it's a gene that codes for one of those proteins. So we can have different versions of the proteins which do different things. For example, we have blue eyes or brown eyes, depending on the proteins that we build. Okay, so an allele is a version of a gene. We would have an allele for blue eyes, we would have an allele for brown eyes, but they're both the gene for eye color. Okay, so that is our alleles. We also need to have a little bit of a reminder of what is homologous chromosomes. Okay, so homologous chromosomes are chromosomes with the same genes. Okay, same genes in the same position. So basically, it's two chromosomes, but they're sort of copies of the same thing. Because you got to remember, okay, we're linking here to sexual reproduction, so we're linking to things where we get one from the mum and one from the dad. So your mum's got a gene for eye color, your dad's got a gene for eye color, so you've got two genes, one from each. And those are homologous chromosomes that those genes are found on. Okay, so chromosomes with the same genes in the same position. So obviously life is a little bit more complicated than one gene for one eye color, okay? But we're going to simplify the world so we can understand it a little bit. So we have our alleles, we have our chromosomes. Right, now we get into a bit more genetic-y stuff. So we need to know what is a genotype versus what is a phenotype. A genotype, well, think about it, genotype, gene type. Okay, so the genotype is the genetic makeup. It's what alleles are present. Okay, now this doesn't always match what we can see. Remember, genetics are all about expressing proteins. So we might not be able to see the visual effects of the genetics. And that's what we're going to get into just now. We're going to have some fun looking at, if my genetics are this, what is my looks? So we have the genotype, but we also, seeing as we can't see that genotype, we need to have something that we can look at to understand how they link together. That is, of course, your phenotype. So your phenotype is your observable features. Okay, so if we just sort of pause here a moment, your genotype, for example, would be your allele for blue eyes, your allele for brown eyes. So it's your allele that you've got for the eye color. Whereas your observable feature is your eye color. What color eyes do you have? Okay, so genotype is the instructions that we've got, which alleles. Phenotype, what we can see. So the other thing we want to link to here is an important set of things. We have homozygous and we have heterozygous. These link to your genotype. Okay, he can be either homozygous or heterozygous. So if you are homozygous, it means you have the same allele twice. So you got blue eyes from mum, blue eyes from dad, you got two blue eyes alleles. If you're heterozygous, you got two different alleles. So you got blue eyes from mum, brown eyes from dad. Both eye color, both the same homologous chromosomes, but different alleles. So this is how we say you have different alleles. Rather than saying full-blown sentences, we say the individual is heterozygous. So homozygous is identical alleles. Oops, if you can spell alleles. Heterozygous is different alleles. Obviously, I'm having problems spelling alleles today. Right, so we now have alleles, chromosomes, genotypes, phenotypes, and homozygous and heterozygous. So let us move on now to the next round of terminology. And that is getting a little bit more understanding of these homozygous and heterozygous and allele virgins. And the thing that we have is, are they dominant or are they recessive? Okay, so we don't have people walking around, also many, with stripy blue, brown, blue, brown, blue, brown, blue, brown all around their eyes. Okay, we do have some funky things going on with eye color because it is more complex than blue or brown. But yes, we don't have stripy eyes. So how does an eye turn out blue or how does an eye turn out brown? This is what this is all about. So a dominant allele is an allele that is expressed if it is present. A recessive allele is only expressed if there is no dominant allele present. And we use capital letters and lowercase letters in order to differentiate between these two. So for example, we would have capital B for brown eyes, little b for blue eyes. So remember that we have homologous chromosomes. So every individual has two alleles. So you could be capital B, capital B. You could be capital B, little b, or little b, little b. And these are your genotypes. Okay, but now the expression comes in the phenotype. So let's have a look at what phenotypes we would get from this. Capital B, capital B. So the allele is going to be expressed is your brown eyes. Because we have that dominant allele present. Twice. Straightforward. Next one. We have a capital B, a little b. Okay, this means we have this capital B, so we're going to get brown eyes. Little b, we are recessive, recessive. So we are going to express the recessive in this case, so we're going to end up with blue eyes. So let's have a link back to this with our homozygous and heterozygous. Here we have homozygous dominant. Here we have homozygous, recessive, and here we have heterozygous. We have different alleles, one dominant and one recessive. So here we have our phenotypes. Okay, so if we now take this, where did I want to go next? Okay, right. So what we are looking here, this is an example of monohybrid inheritance. Now this is a bit of a fancy term, okay? But it isn't, where am I? Sorry. This is a bit of a fancy term, but it isn't something to be freaked out by, okay? So monohybrid inheritance is simply what we're doing, okay? We are looking at the inheritance of one characteristic which is controlled by one pair of alleles. That's all it is. So I've told you already that life is a little bit more complex than this, okay? But we've got to simplify the world in order to be able to understand it. So in order to be able to understand how dominant and recessive and eye color all interlinks, then we simplify the world to our one characteristic eye color controlled by one pair of alleles, brown and blue. Okay, then we can get a grip on things. Okay, and this is then leading us into how do we actually see genetics. So let's get going with that. Okay, so we're not going to sequence DNA and get everybody's sequences and go through the whole works and be sitting in a lab for the rest of our lives. Okay, but there are some simple ways in which we can understand the idea of monohybrid inheritance. Okay, and those two things are Punnett squares and pedigree diagrams. And linked to this, we have the test cross. So, what is a Punnett square? What is a pedigree diagram? And what is a test cross? So, starting with a Punnett square. They are things that look like this. Okay, and then we take the adults. Let's say we have one heterozygous, one homozygous recessive. So then we can fill in what would be the genotypes of the offspring. Okay, so that's one way of looking at things. We can also say these two individuals have offspring, and then we can start filling in, okay, these have a trait and this one has a trait, and then we can start pulling genetics from there. Okay, so we're going to get into each of these in a little bit more detail. I'm not expecting you to know what's going on just by looking at these. I just want to give you an overview first before we get into some of the details. So a test cross is how we actually figure out. So remember, if we look at our genotype and our phenotype, okay, when we're looking at an organism, we can only see its phenotype, but we're not always sure what the genotype is. So, for example, if we had a person with brown eyes, they could be capital B, homozygous dominant, or they could be capital B, little b, heterozygous. If we had a blue-eyed person, they would be little b, homozygous recessive. That's the only way we can get blue eyes. So we're happy with what their genetics are from looking at them. Our brown-eyed person, here we would need to do a test cross in order to figure out which one. Now, I have to admit, we don't tend to do test crosses on people to understand their genetics. What we would normally do test crosses on is plants and mice, okay? But the concept is the same. If you cross an individual, mate an individual with a homozygous recessive, then you can figure out which one of these I've got. Okay, so you cross with homozygous recessive. So we'll have a look at why that works in the next bit as we get into a little bit more on Punnett Squares. Just watching the time as well. So I might slide some of the other stuff I plan to do today into next fortnight's lesson as well. Okay, so in order to see genetics and inheritance, we're going to simplify the world. We're looking at monohybrid inheritance, one characteristic, one set of alleles, two possible alleles. Okay, we have Punnett Squares, pedigree diagrams, and then we can use test crosses alongside this. So let's look at Punnett Squares in more detail. So we have said that Punnett Square is, funnily enough, a square. Okay, so what goes where? Up the top here. Okay, so this would be parent 1. This is their genotype. Okay, this is a parent 1's genotype. Now think again, when we do sexual reproduction, we have haploid and diploid. So the parent cell is diploid, it has 2 sets. Haploid is the gametes. So one will be in one gamete, one in the other gamete. So here are our possible gametes. Okay, then we can do the same thing. Let's say that parent 2 is also heterozygous. Capital B, little b. Okay, so genotype and possible gametes. Then we can say, right, so what are we going to get? What offspring combinations can we get? So we take this B and we pull this B. So this offspring could be homozygous dominant. Bring this B across, bring the little b down. Bring little b and a big B. So it's just one of those things where we normally write the dominant first. Okay, big B, little b is the same as little b, big B. They're both heterozygous. Then little b, homozygous recessive. So all of these are your offspring genotypes. Okay, and that's what a Punnett square gives us. Okay, it gives us what are the possible genotypes for our offspring. And then we can take that from there and we can look at the ratios to understand our cross better. So if we look at the genotypic ratio, what genotypes do we have? We have dominant, homozygous. We have heterozygous, and we have homozygous recessive. We have one of these to two of these to one of these. And when you go through the lesson in more detail, you'll discover that different types of crosses have different predictable ratios in the offspring. So this 1 to 2 to 1 is an indication that we have two heterozygous parents. However, remember that we can't always see the genetics from the phenotype. Our phenotypic ratio, my spelling is awful today, is different. What are the phenotypes we have here? Oh, probably should tell you that we're doing eye color again, hey. Okay, I don't know what happened to the spelling. So B is for brown, and little b is for blue. And you can see the dominant and recessive based on how I've written them. Remember, your dominant is always your capital letter, whereas your recessive is always lowercase. So, now that you've been reminded that we're dealing with eye color, what are our possible phenotypes? If we look in here, we've got some brown eyes going and we've got some blue eyes going. That's all our possible phenotypes. So, brown eyes to blue eyes. How many brown eyes? Remember, dominant is expressed as soon as it is present. So 1, 2, 3 with brown eyes, 2, only 1 with blue eyes. Because if this is our recessive one, it's only going to be expressed if there is no dominant around. So it's only going to be expressed in our homozygous recessive, our little b. As soon as we have a heterozygous, we express the dominant. So our phenotypic ratio is quite different to our genotypic ratio. Okay, but they're both a total out of 4 because we're looking at 4 possible offspring. All right. So hopefully that gives you a good idea of what a Punnett square is. And now we shall touch quickly on pedigree diagrams, and then we shall play with pedigree diagrams next week. Okay, so there's Punnett squares. So here you would have something more like this. And then let's say we would have this. Okay, so what you're looking at here is effectively a family tree. So you have, the squares are used to represent males. The circles are used to represent females. Nice and easy to remember because eggs are round. So we have a male and a female that are crossed, and they have 1, 2, 3, 4 offspring. One of these offspring mates with another male, and then you get 2 offspring. And what there will be in these things is they'll have a characteristic. So they'll shade. For example, this one has the characteristic. This one has the characteristic. This one carries the characteristic, and this one does. And then you've got to work through and you've got to figure out what are the possible genetics. So, some things to bear in mind. Okay? Recessive is only expressed when it is homozygous. Okay, dominant phenotype, no, sorry, it would be the phenotype associated with the dominant allele is expressed for heterozygous or homozygous dominant. So if you see something, let's pretend it's eyes again. If you have blue eyes, you know that the genetics are little b, little b. If they say the individual has brown eyes, it could be heterozygous or BB. And this is the thing. You can only label with certainty your recessive. Your dominant genotype could be either of these. So what is nice to do is to have capital B question mark, because you don't know if it is a recessive or a dominant. And that helps you to keep track of what's going on. So I've actually got some examples from past paper questions of how it works through these, which is a much easier way to explain it by doing. Punnett Squares we can just make up and do, but pedigree diagrams need a little bit more prep, so it's nice to use past papers and see how they put them in there. So we will kick off with that for 2 weeks' time, looking at actually using pedigree diagrams, and then some more of the other funky stuff that's in the guts of genetics and really understanding how alleles and expression can work together to create something that you see versus something that's genetic. Okay, so if you've got any questions, then we didn't end up with much time today, so you can send